Hey y'all. First I just wanted to say I miss you guys. I miss being in lab with you guys, and I hope you're staying healthy and as active as you can during these times. With that, we are going to be talking about the diffusible signals, non-autonomous mechanisms, and how they determine the main proximal distal limb subdivisions. And this is Dirk, me, Brianna, and Maddie. So our first slide, we're going to be talking about kind of what proximal and distal mean and how they relate to the developing limb. So proximal is the closest to the body and distal is the farthest away, and the limbs grow from proximal to distal. Now, the stylopod, zeugopod, and autopod are three different subsections of the limb that are going to be very important for this paper. The stylopod, which is the most proximal, includes the humerus. The zeugopod, which is in the middle, is the ulna and the radius, and the metacarpals and the digits are the autopod, which are the most distal. Now, for this schematic, we're going to be talking about the signaling pathways that help in the development of the limb, and then the threshold and gradients that also contribute. So, starting with the figure on the left, the AER is the apical ectodermal ridge, the AER, and it's a structure that forms from the ectodermal cells at the distal end of each limb bud and acts as a major signaling center to ensure proper development of the limb. So, as you can see in this schematic, the FGF activates CYP26B1, which inhibits retinoic acid. Now, retinoic acid is at the proximal end, and FGF and Wnt are at the distal end. So, retinoic acid is the necessary signal for the proximalizing activities of the cell mites at the proximal end of the developing limb bud. So to continue with this signaling pathway, FGF also inhibits mice and activates HOX13. Retinoic acid activates mice, which inhibits CYP26B1. Very similar to many of the signaling cascades that we've looked at in class. Looking at the figure on the right now, when stained, mice should be seen at the proximal end, the stylopod, because it is activated by retinoic acid. So with high levels of retinoic acid, mice should be able to be seen, as you can see on the left of that graph. Now, HOX should be seen when there are high levels of FGF, because FGF is the one that's activating that HOX13 gene. So this schematic is super similar to the bicoid nanos graph that we've seen before, where the thresholds determine which genes are turned on and how much of that ratio of retinoic acid to FGF is seen and what that ratio looks like. So with all of that, we have seen this pattern all throughout our development studies. Ratio and gradient of two factors, in this case it is retinoic acid and FGF, and their thresholds lead to broad stripes, which lead to narrow stripes, which lead to specialized features. Now, instead of dorsal and ventral axes of a developing embryo, we now have proximal distal axis of limbs. FGF is the distalizing agent and retinoic acid is the proximalizing agent. The further away from the proximal end of the limb develops, the less retinoic acid is present, and the higher the threshold for FGF is. The ratio for retinoic acid to FGF decreases as the limb develops distally. The ratio is high at the proximal end of the limb bud. Within this study, they were trying to determine what signals were proximalizing and distalizing limb development, or if it was simply autonomous signaling. Their background knowledge included the FGF and Wnt signaling, and they chose to transplant the limb buds, recombinant limbs, or RL is what they call them, into the hindbrain because it lacks large amounts of natural or endogenous retinoic acid. So they could determine if the FGF signaling was autonomous or if it was dependent on a gradient like I've kind of already shown you between retinoic acid and FGF. Now remember that retinoic acid helps to form the posterior but not anterior end of the embryo. Without any retinoic acid, they were able to determine FGF could only create the autopod or the distal part of the limb. Thus, the development of the entire limb was not autonomous signaling on FGF's part. That retinoic acid and FGF signaling work together in gradients of two-factor thresholds, which is what we saw in that bicoid in nanos looking graph, in order for entire limbs to develop. and that retinoic acid is necessary for the proximalizing activities of the somites. So proximal distal signaling, PD, is what is studied throughout this pivotal article that made large strides for developmental biology and specifically invertebrate limb development in 2011. So with these take-home messages, Maddi is going to go through Figure 1.